Hey, it's me, Destin. Welcome to Smarter Every Day 2. This is the second channel. On the second channel, what you can expect is a more long-form discussion of the topic that we're researching. So in this particular video, we went to Vanderbilt University. I met Prashant Singh. I also interacted with Dr. Tina Iverson and some other folks that you're going to meet. This is the long-form video that accompanies the main channel video, which is the 300th episode. So Smarter Every Day, episode 300. This is a fantastic discussion. I saw this flagellar motor on the internet. It blew my mind. I wanted to learn more. So I reached out to Prashant in the DMs on Twitter, X, whatever they call it these days. And I was like, hey man, I love this. I would love to talk to you. And we arranged a time. I went up and it was awesome. So we're going to pick up on the campus of Vanderbilt University after I figured out how to park and all that kind of stuff. And we're going to learn about a bacterial flagellar motor, which is fantastic. If you haven't watched the first channel video, that's episode 300, I would recommend it. It gives you the overarching idea of what's going on. But here you go. Enjoy this discussion. I think it's amazing. Oh, one more thing. Thank you to everyone that supports on Patreon because I'm grateful. Yeah, anyway, enjoy the discussion. Hey, it's me, Destin. Welcome back to Smarter Every Day. I am in Nashville and I'm at Vanderbilt University, and I'm here because I saw a research paper online that looked amazing. It's a motor made of molecules. It looks like a mechanical device. I'm an engineer, I'm not a biologist, but I saw this thing and I was like, that's a motor. I want to see how that thing works. So we're here, we're going to find the researcher that's been working on this, and we're going to see what we can learn. Let's go get smarter every day. All right, maybe it's that building? Maybe not, oh no, we're looking for Prash. I think this is the spot. Oh, there you are.
Destin, how are you doing?
Yeah, doing well, nice to meet you. How are you?
Meet you too.
Yeah.
I'm doing well. How was your ride?
Oh, it wasn't bad.
This wasn't bad?
It wasn't bad, yeah. I took the kids to school and then came by. So this is the hospital?
Hospital side, and all of this is hospital as well. Those are some new areas built at Vanderbilt, or new floors built, or kind of more like renovated. But this side is like university side, or the medical school.
Oh, fancy, dude.
So this is Medical Research Building 4. We are going into Robinson Research Building through here.
Okay, cool. Oh, backwards, you're fancy. Do I need to sign in?
We just need a visitor tag for you.
Sounds good.
He's visiting and he will be with me, so just pick up one of the visitor tags.
Thank you.
Thank you.
Look at you got it all set up, ready to go, don't you? You know what's up.
So we are on the 4th floor.
So what is the name of the facility that I'm at right now?
So this is not a facility. These are classrooms for nurses and doctors on this floor.
Okay.
But we'll be going to another building which is connected with this. All of Vanderbilt is very interconnected. We even have like a tunnel system underground which is used to transport patients in case of emergency. So
really?
So we can go all the way from this building to like 4 buildings across. Like you can walk 1.5 miles all the way on the other side of campus in a tunnel. Yeah.
I didn't know that. I've heard about something like that at Disney World.
We call this building Light Hall. And then we are moving into the Robinson Research Building, this is a department of pharmacology. It starts from here.
Okay, so this is...
So it's like...
so what is pharmacology? Forgive the stupid question.
It's just we study drugs, we study diseases and come up with the goal is to come up with drugs that can help solve the problems of human health. And so pharmacology is a department that really tries to get into what the new problem is or an upcoming problem for next decade and try to hire faculties and researchers to solve those problems.
This is where you work?
This is one of the labs we work. So my bench is all the way on that end.
Oh, this is legit, man.
So these are benches, each bench is for a student or postdoc and their equipments, and we can talk more about those equipments as we go. We are also having like a small meeting. Yeah. So
yeah, cool.
This is Brandon. He's a research assistant.
What's up, Brandon? I'm Destin. Nice to meet you, man.
He's Pankaj. He's one of the authors on the paper.
Oh, you're on the paper?
Yeah.
What was your name again?
Pankaj.
Pankaj? Nice to meet you, yeah.
And that's Yvette.
Hey, how's it going? I'm Destin.
Postdoc in the lab.
Yeah.
That's
Jordan, what's up? I'm Destin.
Hi, Tina.
Tina, how's it going?
Good. Keandria.
Keandria?
I'm Caro.
Caro, nice to meet you.
That's my bench. That's where.
This is your bench? This
yeah,
is home?
Yeah, this is home. So when I'm not working on the computer, I'm usually working here doing experiments.
Do you work in the lab with him?
Yes.
Okay, is this your bench? Yeah, this is my bench. So you are bench buddies.
Yeah, exactly.
Can we see the motor?
Yeah.
Let's
come
see
on.
the motor. Okay, so we need to get context here.
Yeah.
So, oh, sorry.
But we can also
There's a lot of smart people walking around.
This is the other.
Is that a human organ?
It is.
Is it, seriously?
Well, a piece of one, a piece of a heart.
Piece of a heart. She's just like carrying an organ around. Okay.
He's probably looking for a lab.
I was joking and I asked her if that was an organ and she said yes. That's not normal. And she was just like, she had her cup. Okay, whatever. That's crazy. All right.
Yeah, it's normal.
That's normal.
That's normal.
Okay.
Please have a seat.
Okay, so we're in the place where Prashant analyzes his data. And just to give us whole context here, you have written a paper with your team in Nature, what's the?
Microbiology.
Nature Microbiology.
Yeah.
And it's about a motor
Yes.
that's made of molecules
Yes.
that's on bacteria.
Yes.
We talked about this briefly earlier. So you were describing that this is a bacterial cell.
Yes.
Is that right?
Yeah, it's a bacteria. Yes.
And you've got a double wall?
Yes.
Okay, so it's almost like a submarine. Forgive me, Prashant. I'm going to say a bunch of engineering terms because I'm an engineer.
I understand.
So it feels like a submarine with the outer hull and then the inner pressure hull. And you've got this motor sticking through the wall, and that motor is spinning, and it's just a flagellum. It's just whipping it
It's
around.
just whipping it around so it can get all the thrust so that bacteria can move forward or it can turn around or
Okay.
tumble.
So this is amazing. And when I saw your imagery on the internet, I was like, man, this is crazy. This looks like a motor that I would interact with. And you explained to me that there is a rotor. There's a little thing that's driving this thing, right? Where's it at?
That is right here. It sits on the membrane, on the inner membrane, and it touches the top of the C ring, which is here in the orange. It interacts with the protein called FlyG, and it turns that protein, as it rotates, it turns that protein, which causes the whole ring to rotate. So what we see here, the two membranes that are here.
What is the outer membrane called?
It's called outer membrane.
Okay, and
And
the
inner membrane is inner membrane.
Okay.
And there's proton filled in here.
Did you say protons?
Yeah, it's filled with protons, hydrogen ions in here. And it's
Hydrogen ions, okay.
yeah. So the hydrogen ions in here fill, and on the inside here, there's very little hydrogen ion. So there's a
Okay.
gradient.
Forgive me, so
Yeah.
we have to go slow for me. So protons, I think of protons as being a positively charged atom.
Yes,
Is that true?
that is true.
Okay, so it's when you say proton, you're meaning an atom that is lacking an electron.
Yes.
Okay.
So this is high concentration of protons in this region.
Okay.
And low concentration of proton in the inside of the bacteria.
Okay.
So now protons, every time there's a gradient, for example, there's a dam, water is up there and there's less water, there's a gradient, energy can be generated, or it could be used, that potential energy could be used to kinetic energy and generate.
So there's a potential difference of electrochemical force of some sort.
Yes.
Okay.
So that's the gradient that this motor uses to turn itself.
Can you show me the video of that working?
Yes. So what you see here is the MOT-AB in brown is rotating, and the motor will rotate in counterclockwise when the MOT-AB is on outside. And the same thing happens again. To move it in the counterclockwise, this protein rotates, and it brings the MOT-AB inside and starts rotating into clockwise.
Okay, so I'm going to get all engineering with you
Yes.
now. So if you have a torque,
Yeah.
you have to have a thing to react against. So that little pinion, I'll call it.
Yeah.
The little, what did you call it?
MOT-AB.
MOT-AB?
MOT-AB.
MOT-AB.
Yeah.
So MOT-AB is this little thing that's driving it.
Yes.
Is it pinned to the sidewall or something? What is
Yes.
it?
It's also in this inner membrane. So it's right here. So there's multiple colors that you see: orange, cream, and green. This all is one MOT-AB.
Okay.
And it takes protons or hydrogen ions from the top and goes into the low gradient here. And as it's doing it, it makes interaction with this red protein here. And this rotates, and it causes the motor to rotate.
Okay, but it's kind of pinned in that
Yes.
wall.
Yes, it is pinned in this membrane, but it can go round and it can shift a little bit in and out.
So it's almost like this is the engine. The Malt AB is the engine.
Yes.
The gas or the whatever would be the equivalent of the activating energy comes through the cell membrane here.
Yeah.
Is this correct?
Yeah, that's correct.
It goes into the Malt AB, and the Malt AB interacts with the red part on the motor, and that's what provides the torque.
That is correct.
That's how it works.
Yes, that's how it works.
Is this called a molecule, or is this
We call it the protein complex. It's a proteins made of proteins, so we call it the protein complex, or we also call it assembly.
So these proteins, are they shaped a certain way that they only
Yes.
lock together like a puzzle, like
Yes,
this?
like Lego. Yeah.
Really?
Yeah. I wish I could show you. I'll show you in a minute, like how they look on the screen, each piece. But when I 3D printed these, it's pretty cool because you can like stack them together and it forms a circle. It was a little hard. You need a flexible material for that, but they can lock into each other and then form.
Yeah, that's
Form
one of
this
my questions, because these, I would assume, are squishy
Yes.
in real life.
Yes, in real life, yes.
So somehow they lock together and they perform. So when you set blocks in a bridge, in an arch, they all would fall apart, but once you get them in a certain structure, they lock
They
together.
locked in. Yes, that's right.
Does this do something similar?
Yeah, so if you see a low resolution structure of this, meaning right now because you see every single amino acid which makes up the protein, you can see every single amino acid, so it's hard to see it, but at a low resolution, this actually is a helical structure. It actually, from the base, it forms a helix. And that helix is what provides the support to this whole thing at the base. And then up here, it's very flexible. The red protein actually moves around between clockwise and counterclockwise. So bottom is the stable piece, top is the flexible, and that is what changes when the motor turns counterclockwise and clockwise. So this is a program called Chimera. It's again an open source software from UCSF. And they make the software where I can put the structures that I've made in here and start visualizing them, understanding what's going on, because this view provides a big picture view.
Will you tell me what I'm looking at?
So what you look there is the protein MS ring right here, the blue one. It is 33 copies of the one protein called FLEF. That is 33 copies of those aligned together forms a ring called the MS ring. M stands for membrane and S stands for soluble. So the reason it is called membrane soluble ring is because half of this ring, this part is in the soluble region, and the remaining part here is in the membrane.
Soluble region?
Soluble.
So in between the two membranes?
Yes, in between the two membranes. So anything that's not in a membrane, we usually would call it is a soluble region, unless it is sticking outside the cell.
Okay.
Yeah.
So so that's just the blue part here. And then what is the name of this again? AO?
That's Fly G.
Oh, yeah, Fly G.
Fly G.
She called it?
Fly M and Fly N.
Fly G, Fly M,
Yes.
and Fly N.
Fly N.
And then this is called what?
Fly F or Fly F.
Fly F.
Yeah.
So 33,
Yes.
approximately 30,
34.
34.
Yes.
And what is this?
34.
34.
And then 34 multiplied by 3.
So why do you have 33 and then 34?
We don't know either. We think that it's a symmetry mismatch, and dynamic complexes like this and ATP synthase that generates ATP, that also is another complex that is symmetry mismatched. Complexes like these, if something is in symmetry, it's stable. And having a symmetry mismatch means the assembly will try towards getting to a perfect state.
It'll try to orient itself?
Orient itself and will constantly work towards it. It doesn't know that it's never going to get that symmetry, but it probably is going to try it, and that's what makes them so dynamic, like turning at 100,000 RPM and then switching again immediately into other motion at 100,000 RPM is like a big change. Something that dynamic happens when there's an imperfection in it. So same thing with ATP synthase, where there's also a symmetry mismatch. We think that the reason there's a symmetry mismatch is what makes this motor running 24/7, like at that, trying to attain that speed.
Can you show me each piece?
Yes, I can.
Okay, so this is the soluble piece?
So this is the MS ring, which is the soluble part is the top, and the lower here is the membrane part, And then this is the C ring, which has 3 proteins, fly G, fly M, fly N, or fly G, fly M, fly N. And that is in soluble region again, and not in the membrane region. Now, if you want to see each one of them, so what you see here is there's more than one fly F, but this is one fly F, how it looks like.
Wow.
So it
Kind of
starts
folded in a really weird way. Yeah.
in a weird way. So if you see this one, it starts here, goes up, and then comes back down.
What is this?
This is single subunit taken. So if you see, this is a difference between the clockwise and counterclockwise. So what you see here is there is no change between the two structures at the bottom, fly N and fly M, but the change is happening in the red part, where it turns, everything turns, and that's the turn that pulls the MOT AB inside. So that's.
So how do you know the structure is in one position or the other? Like when you take these microscope images, how do you know if it was a clockwise or counterclockwise motor?
When we took the first data set, it was always counterclockwise because that's the default state. In the second one, we did mutation, which caused it to get locked into a clockwise state. So we know a mutation, which when you do it, when you delete those residues, it will lock the motor in clockwise state. And that was published in 1986, I believe, or 1980s, 1990s.
Do we know what these inner spines do? Do we know what those do?
Very good question. Those are questions that we are trying to answer. This is like, you're probably the first one who has asked that question. Inside this motor, we think there's export apparatus that exports protein from here to come out and forms the flagellum.
Ah.
So that goes here. So we
like roots of a tree.
Yeah, exactly. So we don't know exactly what their role is in this, because the structure we have is without the export apparatus. That export apparatus probably fell off when we did this. So we have it without the export apparatus. But
what is the flagellum made out of?
It's a protein called flagellin.
I could have named that one.
Yeah, it's a protein called flagellin.
Okay.
So it's just a protein that gets assembled and forms this flagellum.
Does it grow like a hair? Does it have transport of nutrients through the flagellum?
I don't know the answer of that. Does it transport nutrients? But it does build it in pieces, and it starts forming and forms to a certain length. And I don't even know what describes that length, what decides on that length.
So when I think of a flagellum, I think of a sperm cell, a human sperm cell. This is different than that.
This is slightly different.
Okay.
Just because that's human, the origin is human, origin is bacteria.
Okay.
They have similar goals in terms of swimming. They both go counterclockwise and clockwise, but here the purpose is for infection, is for moving from high energy gradient, low energy gradient to high energy gradient, and that has a slightly different morphology and purpose.
The purpose of these flagella is to spin when it receives a trigger,
Yes.
a gradient.
Yes.
So this gray thing out here, is that a trigger of some sort? How does it know when to turn the motor on?
Good question. There are sensors on the outside of the bacteria. So once it knows that there's a threat or there's more energy near me, it senses that, it gets a chemical signal, and there's a cascade of signals that go through, and one of the protein well known for this is called KY, C-H-E and Y, capital Y, KY. That protein comes in and binds to this protein around this region.
Inside the cell?
Inside the cell. And that protein is inside the cell, but at a different location. So the moment it senses that I need to run away from this location, or I want to go to a different location, that protein comes and binds to it. And it encourages the motor to turn in clockwise direction.
Okay, so we need to talk about what you just said.
Yeah.
Because you just created a coordinate system inside the bacteria.
And the bacteria, yeah.
You put sensors on the outside of the bacteria.
Well, it already exists.
Okay, there are sensors on the outside of the bacteria.
Yeah.
Somehow the bacteria knows where a sensor is triggered, and it knows how to trigger what motor on what side of the bacteria.
Yes, and how to turn it.
And which direction to turn it?
Which direction? So that that particular protein will make it go in clockwise. So this is clockwise direction.
Okay.
When it is not attached to it, it will go in counterclockwise. So the default motion is counterclockwise. When the motor turns counterclockwise,
You say counterclockwise from which direction?
from this direction.
From the from the tail,
outside.
the outside. Okay.
Yeah. So counterclockwise would be this,
Okay.
and when the motor is running in counterclockwise, the bacteria will swim forward. So like it's just the boat goes straight.
And that has to do with the shape of if I were to design this,
Yeah.
I would say that would have to do with the shape of the impeller. So the tail?
Yes. So, and I talked about this somewhere else too. It's the way the flagella is made. It's like a whip. So when it starts rotating, it thrusts. The force goes backwards and it moves it forward.
Okay.
Now you would think that it would also do the same thing when it's going in the opposite direction, right?
Yeah.
Now what happens is when it's going in the counterclockwise, there are multiple flagellas on the bacteria body. All of them start forming a bundle.
Yeah.
And multiple propellers form into like one big propeller and pushes this straight, and it goes boom, straight. Now, when it has to it senses like, oh my God, there's a danger. I need to stop. I need to analyze my situation gradient. I need to test. I need my sensors on and test it again. It turns clockwise. When it does that, the bundle opens up, and like in the Matrix movie, there's sentinels. If you have seen those the bugs,
Sentinels,
that come.
yeah.
Sentinels. It's the same thing. There's these bundles open up, and when they open up, it just pauses the whole bacteria, and the bacteria starts tumbling all around.
Starts floating.
360
Yeah.
degree, and
It no longer has a certain. There's got to be a word Latin in here. Taxis.
Chemotaxis. we call it.
Chemotaxis.
Yes, exactly.
Okay.
So we call this whole process of bacteria's mobility like chemotaxis. It's a chemical signal that allows the bacteria to taxis or move from one place to another. And there are some videos that show like bacteria moving from one place to another, and it's just like almost like a video that is like crashing. It's like it's just so fast, but it's a biased random walk, meaning you would look from distance and see like in this plate, and researchers have done this experiment, there's a petri dish,
Yeah.
and they put food in the center and put bacteria on the edges
Yeah.
of it. And you would think the bacteria would go straight to the food, but no, it just goes a little bit straight,
Yeah.
it turns around and goes in the wrong direction, realizes, oh, I'm in the wrong direction, goes back. So as a person, you think random.
I'm getting emotional now
Yeah.
because there's a missile that I've worked on in the past, and it has what we call pulse width modulation control. And so what we do is we take the fins on the side of the missile and we dither them. We go like that.
Uh-huh.
And then all we do is we bias the dithering up or down. We go,
Whoa.
and we can change. So it's constantly moving, but we just bias it just a little bit in order to make a movement. And so what took us a long time
Yeah.
to figure out, you're just describing it. This molecule or this bacteria has an operating system.
Yeah.
It has sensors.
Yeah.
It has effectors. It has
Yes,
actuators.
exactly, yeah.
And it has feedback.
Yeah,
and I'm getting emotional
I know.
because it's so it's a neat design, is what I'll say.
It is, and over the years, this design has like evolved to be so perfect, and as a human, when we look at it, it's like, oh, the bacteria is just running left and right. It's just random, but it's a biased randomness,
Yeah.
and it ends up going to where it's supposed to go. It just takes some time, but it does go where it's supposed to go.
Okay.
And it just figures out. It doesn't have eyes like we do. It figures out by sensing and moving in directions.
It's an emergent behavior based on a few inputs.
Yes.
Emergent behavior is okay.
That's a good term.
Okay,
Now,
let's go back to this. I'm sorry.
yeah.
Just the gravity of what you were describing just blew my mind a little bit. So the structure of this motor, is it well known in the community? Like in all of research, people know about this?
They have seen a low resolution structure. So basically, if I blur this up, they have known that for like 15, 20 years. Like this one looks like maybe like this. So they have known it looks like this blob. There's a ring at the bottom, there's a ring on the top, but what we have is a high resolution structure, meaning we can see each and every amino acid.
So that's what you've done that's so special.
Yeah. Now, why is this motor was so hard for us to solve it for so long? Means we have been solving protein structures for like 40 years. The reason it was because it is embedded into two membranes. And in these two membranes, extracting a complex from that membrane destroys the complex.
Ah.
So it's been very hard to obtain an intact complex for a long time. And with newer technologies, we started going into areas of like new ways to extract proteins, which works for smaller proteins, but something like big, we had to go back to old school purification.
So we've got this double-walled submarine here, and then we know that we have this motor that turns in some way. We couldn't understand the motor for the longest time because anytime we tried to get to the motor, we would pop the membranes and it would kind of fall apart.
It would fall apart because the purification methodology were a little harsh for this kind of complex. So the purification methodology involved sonication, which is like sound waves shooting at the bacteria to break these walls, and what that would do, it would also break the complex.
Okay.
There is something called microfluidizer, which is around 25,000 psi of pressure is used to push these bacteria through a very tiny hole, and they burst.
Okay.
But that burst causes these complexes to fall apart.
So is the integrity of the outer membrane stronger than the bonds of these proteins inside the motor?
It's not, but that force is so high that it ends up rupturing these complexes as well.
Got it.
And sometimes it may not rupture the whole ring, but it ruptures the complex between the blue and the orange, which is the MS ring and the C ring.
The shaft.
The shaft. So what we did was, so when we started this project, we wanted to see how this ring looked like, this motor looked like. So we can't go into a bacteria and take that motor out easily. I mean, there's just so few motors in there. To do this, we have to trick the bacteria into making thousands and millions of motors. So what we do is we use a technique called transformation. We trick the bacteria just the way a virus tricks human beings for like production of more viruses in our body. We do the same thing with the bacteria. What we do is we take a plasmid. So for example, if I want this MS ring, if I want more of those to be produced by bacteria, because bacteria is a factory. So in that factory, if you want more production, I give bacteria that template, like, hey, here's a template. So I give it a DNA that only is for this motor. And I put it inside the bacteria with a process called transformation, bacterial transformation. Once it goes in, the bacteria does not know about it, that a new DNA has come inside it. Then we have a trigger. We call the chemical called IPTG. That chemical is a switch. The moment we add that chemical to the bacteria, it immediately forgets that it has its own DNA. It will start, the DNA that we provided, it will start making that again and again. So instead of having just 2 motors or 6 motors per bacteria, there'll be like hundreds and thousands of motors being made again and again. And
so you're putting the 3D printer file in the bacteria and you're turning on the 3D printer with this, what was the chemical?
Oh yeah, IPTG. Yes, that's a very good to put it.
Okay,
yes.
so you're IPTG?
G.
IPTG.
Yes, IPTG.
So your G code is the DNA.
Yes.
And for the 3D printer, and then you're flipping it on, you're saying run this sequence with the IPTG.
Exactly.
Okay, sounds good. And so you are, how many motors typically does a bacterial wall have in it?
Depends on different types of bacteria. Some bacteria have just 1 motor. Salmonella has motors all across its body. So it could be anywhere, I don't know the exact number, but could be anywhere from like 30 to 100.
Okay.
But what we are doing is overproducing those motors, and they could be in hundreds of thousands.
Okay.
And we grow millions and billions of these cells.
So at this point, I had an opportunity to speak to Dr. Tina Iverson. So we're going to pause with Prosh here, and we're going to go talk to Dr. Iverson, and then we'll come back and we'll continue with Prosh later. Okay, so Dr. Iverson, we're in your lab. You're the PI, do I understand?
Yes, PI stands for Principal Investigator. So that is a person that guides the overall direction of the research and thinks about the research projects in a global way. We might suggest to the different trainees, like, hey, are you interested in this? Are you interested in understanding metabolism and bacterial chemotaxis? And here's how we might go about them. And we give them, the trainees, the graduate students or staff, space to develop their own interests and to develop how they might look at questions so that they stay here only a short period of time and then hopefully go on to run their own labs or research in a different capacity.
So you are the PI, and you are leading all of this research.
Yes.
And I must say, when I was talking to Prosh on the phone, he said so many nice things about you.
That was very kind of him.
He said English is his second language, and you did a lot of work on this paper that got published.
Yeah.
Okay,
Yeah.
that's awesome. So congratulations on getting this published. That's a big deal. And simply put, what have you found here? What has been done?
So we are looking at really this nuts and bolts of how bacteria can move, how they can move toward something that attracts them, like a food source, and how they can move away from something that would kill them, like an antibiotic.
So the bacteria will come along in whatever fluid it's operating in.
Yep.
It'll find a protein and
Yeah.
grab it and then use it like Pac-Man?
Kind of the other way.
Okay.
So the bacteria is moving along. It's trying to find things like sugars or run away from things like antibiotics.
Uh-huh.
And it's either moving toward or something away from something. And then as they are trying to get there, this takes a lot of energy. This is a motor. So it grabs its own metabolic proteins and sort of sticks them on or near the motor to get that motor to run more efficiently. And since we had been looking at the metabolic proteins, we wondered, how does this metabolic protein now act to guide how the bacteria moves? And we were at this intersection and really were able to find this fundamental question: how is metabolism guiding movement? And so we started by saying, like, we'd love to do that, but there's this big black box where no one has gotten at the moving part yet. So let's start here
So
first.
this is like a building block to what you ultimately want to research.
Yes.
Okay, I see. Got
Yep.
it.
So
It's a pretty cool building block.
it is a pretty cool building block.
Yeah.
And we're already in the works for the next step to say, like, now that we have this building block, how do these metabolic proteins integrate into that, and do we see now where this idea comes from that you can have an organism swimming toward or away from something as guided by the metabolism and not just like the sensing.
So that's why you started working on this, because you want to understand this chemotaxis, this moving towards these desirable nutrients. How did you end up here? I mean, this is amazing.
This is awesome, and this is Prash's hard work. He's really been, incredibly diligent about, moving into a new area, bringing some new techniques into the lab. We originally started because we were interested in this intersection between metabolism and how cells work. We were particularly interested in things like cancer, where metabolism changes during cancer. Metabolism changes during aging, neurodegeneration, and a number of diseases.
The needs of the cells change, and so you do different things? Is that why it changes?
Sort of, yeah. And it's actually really not well understood, although it's been known for a long time, that cancer cells do exhibit changes in metabolism.
Okay.
And so we were trying to understand, just at a general level, why does metabolism change what cells do?
And so are you trying to understand how this motor works so that you can figure out what the cells are trying to do? And is that where you're going?
Yeah, I mean, at least for the bacteria, what we're trying to understand is how bringing in energy molecules changes how bacteria swim and how they reach either other nutrients like sugars or run away from other foods. And in the bacteria, and what our next step is actually proteins that support metabolism become physically part of this motor. And this motor grabs them from the bacteria and says, like, hey, I need to turn this way or another way. And it may be part of bringing more energy into this motor, because this motor uses a lot of energy.
So I'm a mechanical engineer, and when I'm doing the camera thing, when I think about how a motor runs, I'm thinking about voltage, armature voltage, electromotive force. What's making this go?
It is electromotive force, actually.
Is it really?
It is, yeah. So there are two membranes here. This one, and our experiment removed the membrane, so we don't see it. But there's one here, and it has a whole lot of proton motive force across it. Some say that it may be as much or more than what's observed in a lightning strike.
Are you serious?
Yeah, it's a huge amount that's across the membrane. And this little stator protein, this MOT-AB,
This little pinion thing.
this little pinion thing that can move in and outside, actually takes protons from one side, and as it pulls it down, it starts turning.
So it's not actually touching. So
They're hydrogen bonding, for example. So they are, at a molecular level, things don't ever like really touch, but they might be just a couple of angstroms away from each other.
Okay.
And making a chemical interaction at that level.
So this little pinion you were describing, this Mote
Yep.
AB, it takes the proton energy in, or the hydrogen ions, right?
Yep.
And then it uses them.
Yep.
What does it do when it uses them?
So when it uses them, it takes them from a region where those protons are very concentrated. Protons are positively charged, they repel each other. They're going to want to have more space down, and they're going to move to somewhere where they're not so concentrated, and that provides power.
Got it. And so that power is then used by the, what is it called, FlyG?
Yep.
That's a good name. Like,
Yeah.
I'm gonna call it the fly gear.
All right. It is. It's the fly gear.
Yeah.
It absolutely is.
So it's using that, and that interaction happens there, and then it's actually providing torque.
Yes.
And then we've got slip from the flagellar rod here. What is this seal here, this pink thing?
That's called the LP ring, and that structure was done not by our group, but Prosh was able to add this to his model because it was defined by two different groups about 2 years ago.
So I'm understanding a little bit. So on a motor, like an electric motor, we
Yeah.
have a stator, which is the static part, and we have
Yep.
the rotor.
Yep.
You guys have modeled the rotor for the first time. Is that?
Yes, so we've modeled this rotor here, and it's the gear that reverses. It also helps, it can interact, and actually this little brown thing is called the stator.
Is it really?
Yeah, it is.
Okay, yeah.
So it can interact, I'm showing it here with one stator, but it can interact with up to 11 stators to get more and more torque put in, either if you want to swim faster, or if you're in liquid that's more viscous
Yeah.
or harder to swim in. So then you can add more and more torque just to keep that same pace up.
But if you do that, you're burning your hydrogen ions faster.
Yes, you are.
Okay.
And then you want more of those metabolism proteins near there. And one of the things those metabolic proteins can do is help boost that gradient, that proton motive force.
I see the matrix now. So you're studying the
Yeah.
whole system. So Dr. Iverson, question. If we have the outer wall of the bacteria and the inner wall of the bacteria, how is there a seal right there? Because one of the big things in engineering is, I'm thinking about the space station, for example.
Yeah.
They have this knob that has a rod going through the wall of the space station. They have to have an O-ring in between. Is it an issue when you're this small? Can fluid go through that?
That's a great question, and the answer is semi-no, and part of the answer actually comes into a part that isn't shown in this figure. So we don't show the membrane, which is here on this blue region, and up here is the peptidoglycan or outer membrane layer. There's also a region of this Mote AB that should be connecting up here to help stabilize the whole thing. But as we think about, let's say, a water molecule, and how big that is, when the membranes are up against this, and those membranes are very hydrophobic, they seal up and they don't have enough space for that water molecule to go through unless you've got a specific channel or pore that the bacteria puts in.
So we're operating at a level where you don't have to seal things because the molecules of water are so big. Is that right?
Sort of.
Not really. Okay.
Sort of, but yeah, I mean, the actual membrane themselves are other types of molecules that are packed together in a way that wouldn't have a hole big enough for that water to go through, and they would repel that water.
But the design accommodates for that seal in some way that, as an engineer, I just don't quite understand.
Yeah, no, I get that. And one of the things that's challenging here is that when we looked at these molecular components, we actually pulled them out of the membrane. So that's a part that we didn't get to image here. And so we don't get to see the seal and how that seal might be adapting around any motion that we see in this motor. One of the ways the entire field is going now is, there's been this ability to image very small things with fine detail, but sort of medium and larger things in the cell with more blob-like characteristics. Oh, okay. And some of the new technologies are now getting to these larger assemblies of proteins at finer details.
So we're seeing the overall system.
Yes. So before we were putting the system together from component parts, and now we're seeing the system more and more intact. And there's these bridges between the molecules at an individual level, which can tell us a lot about how they work. But molecules together working in concert tells us much, much more.
So you said you're a physicist by training.
A chemist.
Chemist. Okay, and so you're running a biology lab kind of thing here.
Yeah.
Is that the way it works? I don't know this world.
We're a pharmacology department, and I'm in pharmacology and biochemistry. I did my doctoral work in a lab in the Department of Chemistry.
Okay.
Also looking at protein imaging. But this has been, it's a really, I think bringing physicists and chemists into biology is really important and key for the field, because people are trained to think a little bit differently. And the bacterial chemotaxis system was actually originally looked at a lot by physicists, not exclusively, but you can see aspects of how the field developed that is really influenced by how physicists think.
Interesting.
That really thinking about how probabilities affect which way you're turning is really from the physicist mindset.
The bias,
Yep.
What was it called? Random bias?
Yes, the biased random walk.
Biased random walk. So can you briefly explain biased random walk so I understand it?
Yeah, so you've got a bacterium that swims straight. Their flagella are going to come out of their little bacterial butts.
Okay.
So they have more flagella on one side, but they're all turning the same way.
Okay.
They're all turning the same way. They're all turning counterclockwise, and the bacteria go straight.
Okay.
When you turn clockwise, the flagellar bundle unwinds and the bacteria changes direction.
Okay.
If you take the flagella out of the bacteria, they go straight 100% of the time.
Okay.
But if they're in the bacteria, they go straight and counterclockwise and straight 70% of the time, and 30% of the time they turn the other way.
Say that again now? If you put the bacteria...
If you have a whole bacteria, it swims straight 70% of the time. If you take the bacteria and cleave off just the flagellum part, so the bacteria's had its flagellum pulled out.
It's like a half of a bacteria.
Yeah, it's no longer a bacteria. It goes straight 100% of the time.
So it's something about having the whole system that changes it to 70% instead of 100%.
Yes.
And we don't know what that is yet.
That is actually what's called a signaling protein, and these are proteins that bind to it, potentially somewhat stochastically. It'll have a certain probability, and this is where those physicists come in. There's this probability that it might change when you have things that are turned on at a low level. Mm-hmm. So you've got some, bacteria are alive, they have something that's on at a low level, and when it's at a very low concentration, it sometimes flips it. And then that signaling protein, when you've got glucose over there, you're moving toward, that concentration is increasing, and it might encounter this more frequently. And so it's going to start to flip it more frequently.
Got it. But if it's spinning in one direction more often than the other, then it's going to give an overall bias to the whole bacteria.
Yep, and it's based on sort of frequency of interactions over time.
Got it.
So that's where the physicists came in and influenced this field to think about bacterial movement.
So if I have just a field here, and I have more glucose here than here, mm-hmm. and I just drop one of these bacteria in there,
yep.
How does the bacteria move towards the glucose?
That's a great question, and that's how this all came about, this biased random walk, which is a bacteria is so small that if there's more glucose over here than there is over here, the front to the back of the bacteria can never sense a difference in more glucose over here than over here. So what it does is it spends time going in different directions.
Really?
Until it figures out I'm over here. So if it'll spend a little time going this way and it says, oh, there's less glucose. A little time over this way, oh, there's more glucose. A little time, and I'm imparting more sentience on the bacterium than probably I ought.
Okay.
But I think it's easier to think of it that way.
But it's like a procedural thing.
Yes.
So there's certain rules that the bacteria has.
Yep.
And those rules end up giving it this emergent behavior where it ends up in a certain area.
Yep, and sort of floats around until it gets to the highest concentration, and that's where having some sort of random probability, it's not that random, probability of switching back and forth really helps the bacterium.
So it's not like something that thinks.
No.
It's a set of rules that ends up giving it a certain preference for a certain state.
Yes.
Is that a good way of saying it?
I think that's fair.
Okay, that's amazing. So this is what you're studying, and the goal is if we can influence these motors, maybe we could put something in the way of the bacteria that changes the way it behaves.
Yeah.
Is that what we're going for?
Yeah. So, I mean, part of this is just understanding how does any organism respond? So that's a really basic fundamental question. What that gets at in bacteria is how do we change how it responds? This is important, it's not important, it's required for infection. E. coli, Salmonella are highly related in their motor. And E. coli infections are the number one, to the best of my knowledge, antibiotic cost out there, and you get recurrent infections. And so can you find ways to make antibiotics more potent, or can you find ways to decrease the recurrence of infections by having the bacteria not hide?
Why are you excited about this?
Why am I excited? Because it's cool.
It is, isn't it?
So first of all, it's just there's aspects of science that I think are just really, really cool, and this is one of them. It's like we get to this nano machine, and sometimes when you get into how a molecule works, it gets very technical, and it's like experts love this, and this is like we get there, and it's like a motor that looks like a motor, and it's just extraordinarily cool to see how you might work this. There were questions that we had before we started this that were like, we didn't know how to answer them, and now they seem so simple.
Really?
Yeah, like, we had talked a minute ago about the proton motive force. It pushes down one way, but it runs a reversible motor. And in biology, if you have, things going one way, when you reverse it, it should go the other way. You shouldn't have things going one way and have it be reversible. And that's why that movement of that FlyG protein allows it to run a reversible motor. But we had no idea. We knew FlyG had a different conformation, but it was not really clear how you could run a reversible motor from a single unidirectional proton motive force, from a current going in one direction.
So do we think, can I use your slide for a second?
Oh, yeah.
So do we think that the FlyG is moving in diameter, or do we think it's moving, how do we think it's actually changing the use of that MOT-AB?
You can kind of see as it goes between states. So we're going to stop it. That FlyG changes, and there's another movie I'll show you if you let me.
Yeah.
All right, so here we have it, a clockwise rotation, and then we're going to strip down to one subunit, and you can see this motion where we have very big changes, and that turns around 180 degrees. That's going to move that MOT-AB from turning it clockwise to
So it's
counterclockwise.
almost like a gear. It's almost like the gear can shift a little bit and become an internal versus an external.
Yeah, it's like if you had a gear and it had spokes on the outside, and then you just turn them to the inside, and it brings that stator with it.
It brings it over.
Yep.
That's amazing. And so how do they all change all the way around the ring at the same time? Is it like a domino?
So that is like a domino effect. These proteins are actually interlaced.
Yeah.
Parts of this look like a split lock washer, if you're familiar with that. And these parts at the top we would call it intercalated, where they are interlaced between, so when one moves, they push the other. But they would have these binding proteins I've been talking about. It could either be a signaling protein or a metabolic protein that pushes on this, and that's where we're going next. And that's
so that's the next thing you're going to look at.
Yeah, that's we're doing preps on that today.
Oh, that's amazing. Who's funding this research, Dr. Iverson?
National Institutes of Health.
Okay.
Has funded this in the past and hopefully in the future.
It's if nothing else, they have really good things to show for their money, right?
Yeah.
That's awesome. This is incredible. If I were to ask you, there's people that may end up watching this video. There's somebody out there that has like command of budget to study this stuff. What are you hoping somebody would see about this and be interested in and supporting?
Oh, there are so many cool things here. So, part of this is this specific project looks at basic mechanisms for how bacteria infect. And that can give us a lot of information on how to develop new therapeutics. And, one of the things we're doing right now is trying to get this down to a little more fine detail to push ourselves there. Antibiotic development is something that's really underdone in the US because it's not very profitable. And I think that in the long term, combination of public funding may be really important to develop these. And so when we hear things about the antibiotic crisis in the US or in the world, where more and more organisms are becoming resistant, it's because there's just not enough done to develop new antibiotics. So as we understand the molecules behind processes in bacteria that are required for infection, we can help think of new ways to help either complement or provide new sources of antibiotics, as well as new ways to prevent bacteria from forming reservoirs that result in the recurrent infections that are important for resistance. So not everything that we look at in bacteria will end up being able to be this target, but we don't know until we try, until we look at all the different ways, how does an infection start, which ones are going to be good targets. And that publicly funded research is really important for that.
So you're probing into the unknown.
We are probing into the unknown. At the same time, we found a really cool molecular motor that, we have lots of other thoughts about how to co-opt this for other nefarious purposes of our own.
So I see this, and it's compelling to me because I'm an engineer and I can understand turny things that make things move. It's compelling. And so that's why an engineer got in the van and drove up here to talk to you.
Yeah.
But you're thinking about this on a totally different level. You're thinking like, how can I use this mechanism to help people in the long run?
Yes.
Okay. So you're
Yeah, and we love the turny thing too. We don't get me wrong. We love the turny thing. We think it's awesome. It is. It's really, really compelling to look at.
It is.
Yeah.
Dr. Iverson, thank you so much.
Oh, thank you for coming. This was a long drive, and I appreciate you coming up.
Yeah, thank you so much.
Yeah.
All right, so where are we going?
So we're going to the Cryo-EM facility, which is in the new engineering building.
Cryo-EM.
Cryo-EM.
Cryo meaning very cold.
Yeah, electron microscopy.
Okay.
So we use electron beams to see. So for example, in the light microscope, we use light to do to check the images. In this case, we are using the beam of electron, which is being imparted by a source which is at 300,000 volts.
Okay.
So it's a very high energy beam that shoots our sample, but these samples are frozen in liquid ethane, so they are vitrified. So this is a new engineering building. This is the old one, Olin Hall School of Engineering. They built this new building, so we'll go all the way down in the basement.
Oh, it is pretty. Hey, how's it going?
Hey, good, how are you?
I'm Destin, nice to meet you.
Cool, yeah, I saw your videos.
Oh, cool, yeah, thank you so much, man. Pleasure to meet you.
I like the stairs because, depending on how tall you are, you can choose your radius.
Oh, yeah.
So some people might go on this one, while others may go further left. Hey, guys.
Hey, how's it going? Hey, I'm Destin.
Miriam.
Miriam, nice to meet you.
Scott.
Scott, nice to meet you. How you guys doing? Yeah. All right, cool. Thank you very much.
All right.
All right, we'll be around.
Okay, sounds good.
Sweet.
I was watching your video shooting the 22 trying to light a match, and I was like, this is right up my alley.
Yeah, right?
Like, we're going to come back tomorrow and get this right. I'm like, yes, I like this, yeah.
So, Miriam, you went to MIT?
No, that's just a cup. No, I went to, for undergrad, I went to Bryn Mawr College, which is a women's college out in Philly. And then for grad school, I went to the University of Glasgow in Scotland.
Scotland, yeah, that's nice.
And did my master's out there.
She's our tomography expert, which is one of the techniques that we do, and the process trying to get into next. We can all do it, but Miriam's got a special niche for it.
That's your thing.
I did some like structural virology when I was at Glasgow, and we did tomography like collection and a lot of data analysis, which was pretty fun.
So now we're with Scott and Miriam, and these are the imaging experts. Am I saying that correctly?
Sure.
Yeah.
Okay. Yeah, does that work?
Absolutely.
Yeah, I think we're
And
do a pretty decent job.
We're going to learn about cryo-EM,
Correct.
which cryo means cold.
Cryo means cold. That's like minus 170 degrees Celsius.
What's the fluid that you use, nitrogen or?
Yeah, everything's under high vacuum. So once it's in a microscope, it's under high vacuum, but it's all cooled by liquid nitrogen as well to kind of help maintain that high vacuum.
Okay.
All right, so it's a little messy in here, but we'll talk about how the space was made first.
Yeah.
So when we inherited this space, this part of the building was empty. So what was actually, we affectionately called it the pit space.
The pit space?
The pit. So it was this large empty space.
Legit. Okay.
And we don't know what it was designed for, but it was here. And so we got access to the space. So we built 3 large microscope rooms in this wing.
Is this bedrock?
There's bedrock underneath this, yes.
Okay.
And then it's just the lowest part of the building. We're about 40 feet below grade. And then so each room has 4 concrete block walls. And they're all separated by an air gap between each of the rooms.
So we're mechanically isolated here.
We're isolated. Each of the wall, they're backfilled with concrete, and they have a 9.5-inch pad of concrete on top.
Okay.
So like we are like a little mini bunker inside the building.
This is Tornado, this is where you come.
Yeah, yeah. So every single microscope room is built that way.
Okay.
And because we had this giant hole, we had the option of either backfilling the hole, or we put everything on an elevated platform. And so we decided to put things on an elevated platform because that gave us like ultimate flexibility for microscopes that potentially got bigger as time goes on. So that's kind of happened. So this is our middle room here is our glacis microscope. So we'll walk in and look at it.
Glacis?
Glacis.
Is that an adjective? What does that mean? Or is that a brand?
So they're all code names. So the cryo-EM microscope that we used to have was called the Polera. So polar, cold. These are all made in Eindhoven and the Netherlands. And so the big microscope we have is the Titan Krios. And then this is the Glacis. So they all have a cold sounding name.
Yeah.
Okay, cool. So this is just the vibe. We're going
the
for
vibe.
the cold vibe?
Yeah,
It's
we're
the
going
cold
cold vibe.
So,
and when everything's in liquid nitrogen, you got to be cold.
So this is the Glacis. This is our screening microscope.
I'm sorry, what word did you say?
Screening.
What does that mean? It's like the course measurement or like?
What it really means is you have like a wild range of optimization that needs to be done. So for users, so then they have to screen their samples until they have something really nice that we can collect on.
We're trying to find out what's good and what's not good. So this microscope is it's the lower end. It can collect really great data. But it is not quite as stable and not quite as optimized as our big microscope is.
So this is like a quick look.
Just a quick look. Our samples go in this little thing. We'll show you what the samples look like in a minute, but they go in here, they get loaded in here, this nice dark hole where you can't really see anything.
Okay.
And then somebody will always ask, what is the foam cup for?
No, I didn't care about the cup at all.
This is our emergency off switch. But we just keep it accessible.
So how many microscopes do you have here?
So in this building that we operate, there's 2.
Okay.
We just passed an engineering microscope that is something that we don't operate. Next door to us, there's 2 microscopes. There's a cryoclim, which is a quart of light electron. It's a confocal microscope, a much lower resolution microscope. And then there's a focused ion beam scanning electron microscope. There's 2 of those also. What we look at is transmission electron microscopy. So we have a source at the top that transmits an electron beam all the way through the column, and we put our sample in the middle and a detector at the bottom.
So you're shooting through it.
We're shooting transmission. We are going all the way through that sample.
So it doesn't have to be conductive then?
No.
In fact, we're putting it in that fluid.
So we take a sample that's in a buffer or water essentially, and then we freeze it onto a grid, and we'll show you what those look like.
Okay. What's the fluid, Miriam?
It's frozen in liquid ethane, but everything gets cooled down in liquid nitrogen.
Now you said frozen in liquid ethane. Frozen normally means solid, so how do you freeze something in the liquid?
So you have whatever sample you have that's in a buffer. It will get plunge frozen into, and we have one of our machines right there, which is like a GP2 that can be used for plunge freezing. And you literally just drop your sample into liquid ethane and just gets flash frozen. So your sample is in vitreous ice then, and that is helpful for like near nature.
Vitreous ice is a concept that's vitally important to cryo-EM. So we're freezing the liquid so fast that it doesn't form ice crystals.
Is it just one single crystal?
So it is a transparent sheet of ice. So it doesn't have crystalline ice in it. So if we actually get crystalline ice, that makes the beam not be able to pass through the sample anymore. And it causes a bunch of artifacts. So we don't want crystalline ice. So we want this vitreous ice, which is a very specific layer of ice that's not crystalline.
Okay, got it.
All right, so all of our sample goes onto a grid. So this is a copper grid that has a...
Am I looking at a disc with a rim on it?
So this is in a special holder that goes into what we call our autoloader system on this microscope. So the autoloader system is shared between the Glacius and the Krios microscope. And so a grid that we put in here and clip into here goes into the microscope. It allows the microscope to, this clipping mechanism allows it to have a more robust way of picking up and loading a sample without breaking it.
Oh, I see.
And so then that can be exchanged. And we've had these samples shipped across the country, across the world, and they make it. And they're transferable. Frozen. They're transferable between microscopes all across the world. There are hundreds of these, so it's a very easy mechanism to have the sample just make it wherever you need to make it.
It's like a cassette tape. You make it into you package it so that it can be sent somewhere else.
Yeah.
Okay, so that's what does it look like? Is it a grid?
Yeah. So
it's a mesh work on there.
Yeah.
What's it made out of?
Copper.
So they can be made out of copper, gold. There's some other materials that we use for other very niche purposes.
Okay.
But most of them are copper. The mesh is a support network that then holds typically a sheet of carbon with little holes punched in it. And that sheet of carbon, we want the vitreous ice to form in the holes of this piece of carbon, and we shoot through the holes.
Got it.
So we're just making a network that can hold little tiny sheets of ice with protein trapped in it.
Okay, got it.
We're not going to turn it on right now, but this is a dry room. It looks like a cold room.
Okay.
But we have a lot of high humidity here in Nashville, so we like to have a dry room. It's one of these kind of luxuries we have here on site. So I can show you more specifically here. So we have,
To be clear, what is our overall goal here? What are we doing?
So what we're trying to do is we're trying to trap proteins in a native-like ice layer or buffer state, so that we'll take thousands and thousands of images of these particular proteins, and then we'll use some computer software to computationally, to translationally, rotationally align all these proteins together to get a high resolution structure. Most of the work that we do on campus is directly to go at human health and disease. So we want to understand what proteins look like from our body or potentially from bacteria that are harmful to us, so we can understand what they're doing, so we can understand how to either medically intervene.
I see. So we're trying to take pictures of those things so that we can understand them.
Yes.
And you are trying to prepare the sample so that we can take the picture.
Correct.
Okay, got it.
Yeah. All right. So this is the bottom. This is a clip ring. I always get them confused. So this goes into this particular device. This is our clipping station. Can I have one a grid, please?
What does the verb clip mean, to clip something?
Well, we will show you here. So what we're going to do is we're going to sandwich this grid onto this device that allows us to hold it. So this grid is just a copper grid, again, and it's very fragile.
Okay.
So we want to put it into this mechanism that allows it to be a little bit more robust, so we can manipulate it a little bit easier.
Okay.
All right, so we're going to put that into here.
Miriam, did you see me change between those two cameras like that? I was
Yeah.
I was impressed with myself.
Typically, this is done under liquid nitrogen so that everything stays frozen.
So you're putting that there. So we're loading the sample. There would be a sample there, or you're about to make a sample?
There would be sample there.
Yeah,
Okay.
The sample would already be on the grid in a vitrification machine, which is called there's a number of different ones.
Okay. So what are you doing now?
So now we're going to put this
C-clip.
C-clip on top. So this is what holds the grid into the clip ring. So now,
I'm sorry, you're using a lot of terms that I don't know yet.
Yeah.
The grid is a little copper thing.
yeah. So and then we're putting the grid on top of a clip ring.
Those are the clip rings.
Those are the clip rings. And then we put the grid on top of one of those.
Okay.
And then we put this little spring on top, which is called a clip. So they're all the same name. I didn't name these. This was the way the company came up with it.
Come on, man, get it together.
Yeah, you know, I have to deal with that. That's why I was reading what it is, because I always get it confused too. So this is essentially what we're doing is we're just kind of putting this little tiny spring on top of the grid to kind of get it to hold into this device now.
Got it.
All right, so now you can see that it's all sitting in here.
Uh-huh. And so that device is all together now.
That's all together, and that's what goes into the microscope. What the machines that we have, we typically put the sample onto the grid first. We keep it frozen. We put it into this clipping station, frozen. Everything will be in liquid nitrogen. And then we clip the grid frozen in liquid nitrogen. And then we would store it in liquid nitrogen. Everything from, once you add the sample to the grid and you vitrify the sample, everything stays in liquid nitrogen until it gets into the microscope.
So this would be under liquid nitrogen.
It's completely under liquid nitrogen. Yeah.
Yeah, that's a lot more exciting. But yeah, I'm not saying this isn't exciting.
Yeah, it's harder to see because there's a lot of vapor that comes off, so it's harder to.
My point is, it's a complicated thing.
It's a very complicated thing.
Okay.
So this is
Is
one
it
of
a
our,
microtome?
No,
Okay.
but you're getting at the same idea. So all of our equipment is kind of made by the similar manufacturers. So a lot of the microtomes are made by Leica, so you can see that. So this is a Leica GP2. So this is a machine that does our vitrification for us. Turn it on here.
Mm-hmm.
So what this machine does is that it holds a set of tweezers, and we can walk through this process. It holds a set of tweezers. It allows us to add sample to the tweezers, and then this is a blotting machine. So it takes a piece of filter paper, and it touches the grid very softly, and it actually will wick the liquid off. So we add between 2 and 3 microliters of sample, and we want 2 to 3 picoliters of sample left on the grid.
Wow.
So we're taking off the majority of sample.
So this is a subtractive process here.
Yes, and it's a very unreliable process, to be honest.
Okay.
So this is a blotting plunger. We have a different version of this across campus.
So this plunger device is pretty interesting, and I know the words that Scott's saying, but I don't understand how it works. So when I got home, I asked Prosh if he could give me some B-roll of how these things work. Now, Scott's describing a device that's made by Leica, and Prosh shot some video on one made by Thermo Fisher, but you get the idea. They take the sample and they plunge it down. So I'll just let some of this footage roll so you can see what happens. It's pretty interesting. Okay, I thought that was pretty cool. Anyway, we'll go back to Scott and let him explain more about it.
There are new pieces of equipment that are significantly more expensive that are blotless plungers. And so instead of putting on a bunch of sample and removing it, they only add the amount of sample that you need.
So so they're additive. You're fighting capillary action here. Is that right?
Yeah, to some degree, yeah.
Of liquid nitrogen.
Mm-hmm.
Well, what we're doing
of
is
your sample,
we're adding the sample. We want it to spread across the grid, but then we're removing. We're trying to get a uniform. And so I'll show you what that looks like on the microscope.
Okay.
I think that it might make more sense to see what it looks like on a microscope.
Okay.
All right. So,
Sorry for my dumb questions.
No, it's not dumb questions at all.
All good.
All right. So this will give you a better idea of what a grid is. So we put the sample in the microscope. This grid is frozen. It's sitting in the microscope, and we've done what we call atlasing. And you can see here the overall grid meshwork. Mm-hmm. And then if I zoom in, So now we see that there's this carbon mesh that's laying over the top of the entire grid, and there's little holes in the carbon mesh.
Oh, wow.
So in those holes, we're hoping to freeze the sample in those holes. And at this really high level, this particular grid square is what we call this, looks very dirty. There's a lot of kind of background black stuff. But we see these more white areas here are empty holes. And then the ones that have a little bit more dark in them actually have ice in the holes.
So is this something we did with our screening microscope?
Yeah, so this is in the screening microscope.
So this is like our first look?
Yes.
Yeah.
And so what somebody wants to do here is they'll put in a sample, and if their ice is too thick, the whole grid will be black. So they'll say, okay, that one's trash. Let's just go with a different sample. And you can get an idea if I change samples. So this is something that was already recorded by a different user today. All right, so this particular sample was made on a vitrification machine called a Vitrobot Mark II or Mark IV. And so typically what happens is that you have the tweezers that hold the grid as you're trying to blot off sample. And that most likely was on this edge. And we have a gradient of ice where you can see this ice down here is very thick. So it's so thick that the electron beam can't transmit through it. And then we have a gradient across the grid. Mm-hmm. And so you can see if I zoom in here, these squares are very thick ice and have very few holes. Right? And then if you go here...
So that means the sample wasn't prepared well in that area.
In that particular area.
Okay.
And so the advantage of this particular plunging machine is that you get a gradient. And so sometimes your sample looks better in thick ice versus thin ice. Sometimes you have a very large sample, so you might need thicker ice than you do a smaller sample. Again, in the screening process, what we're looking for is a grid that has uniform ice or has a number of spaces that we can image thousands of times. And this microscope is quite capable of collecting a high resolution data set. And what we mean by that is that we want to see specific features of a protein after we do our data processing that allow us to understand more about it. So some people, what they consider high resolution is different. For our particular field, 3 angstrom resolution is what we consider to be high resolution, and anything higher than that is very high resolution.
That's tiny.
And so you're able to distinguish things that are within 3 angstroms of each other, so at that point we can see what the protein looks like. We can get an idea of what secondary structure is, and then we can also get an idea of like what the amino acids look like on the secondary structure of a protein.
In a blobby level or like a picture level? Like at 3 angstrom resolution, will you know what the coil looks like? Or
Absolutely.
You will?
Yeah, so we can see alpha helices around 8 angstrom.
I'm sorry, what?
An alpha helix, like the coil of the protein.
Okay.
So beta sheets is another secondary structure of proteins you can start to see around 4 angstrom. And then, so when you get in that 3 angstrom, you're high enough resolution that you can start to understand how that particular protein is made.
Mechanically. What it looks like, folded wise?
Yes.
Okay.
Yes.
Okay.
Yes.
Gotcha. And so we're making a grid. We're trying to get a thin enough sheet of ice so that we can measure a sample and we can shoot through the ice.
Yeah.
And we're hoping that there's a sample there that we can hit. And if we can hit it thousands of times, then we can build this blobby picture, and then we can go back and we can put it all together and we can make a 3D model.
Correct.
Do I have it?
I think you have it.
Okay.
I absolutely think you have it. Now let's go to the big microscope where we actually see the insides.
Wow, it looks cool.
Yeah.
So you just waved at the light sensor?
I did.
Oh, that's fine.
Just so
staying
that
here?
it would turn around.
Oh, we staying here?
Yeah.
Okay.
So this is our Titan Krios. This is a Titan Krios G4, which is, I think, still the newest model available. So these doors open where the Glacius doesn't open, but this is the high-end microscope.
That's what you were wanting to do, is the reveal, weren't you?
Yeah, the grand reveal.
Okay.
It's the best part of the tour.
It's the best part of the tour.
Yeah. So our electron source is up here at the top.
Okay.
So it's a 300 kilovolt electron source. Our sample, that little tiny grid, goes into this L-shaped mechanism here called the autoloader.
Okay.
And that automatically transfers the grid in and out of the microscope. So this is the this kind of rough cylinder shape here is the column, and that little tiny sample goes inside the column. And the electron beam is very, very tiny. So we have this massive machine that's looking at this very, very small piece of the grid, and that grid can be imaged. If the whole grid was perfect, you could image it for weeks. I mean, the magnification that we image at is so high. You can do you could cover a credible amount of area. We imaged for a user this weekend and got 40,000 images, and that was not even the whole grid.
And so is there a multi-axis stage in there that moves it in X, Y, Z?
X, Y, Z, and it also has an alpha tilt.
Okay, got it. And so you're able to, it's a transmission electron microscope. So you're shooting through the sample,
Yes.
which means you have a detector on the bottom.
Yes,
Which
that's
is
the next step here. So this is our sample on the bottom here.
Okay.
So we have a detector on each side here on the left and right, and then we have our big detector, which is a Gatan K3 detector on the bottom.
Okay. So you are shooting through and then you're detecting something. And so what are you detecting?
We're detecting the electrons that transmit through the sample.
So one other question I have is, so we loaded up these grids, we went through the screening microscope, we figured out which grids we're most interested in on the grid square. We put them in the autoloader. This is an electron beam, so this is in vacuum.
Correct.
You have to put it in, not in vacuum, and it has to go to vacuum. So does it take an amount of time to pump down?
Yes. There's an automated system where we put the sample in liquid nitrogen in a little canister, put it in the microscope, it pulls up to it, it vents the autoloader, it grabs the sample, and then pulls vacuum very quickly. Okay. So it has a turbo pump that pulls on this autoloader. Once it gets to high vacuum, then it can exchange with the microscope. So there's a number of different valves and pumps along this process that keep everything at high vacuum and keep everything kind of sealed off from each other until it need be.
Okay. So I have a question for you, Miriam.
Yes.
So you said, I heard the word tomography,
Yes.
and I think I know what tomography means, but is that what you do with the data here? What is tomography?
So some of the data, so a lot of the collections that we do is single particle analysis. So we're just looking at one protein. That's like 1,000 of them in one image, and then you get thousands and thousands of those images. For tomography, you're looking for like a whole cell protein or whole cells or viruses, and where we have that alpha tilt, you're actually collecting a tilt collection, and that data gets compiled together and you get a 3D volume of your sample. You can then answer questions of what the whole entire cell can look like, or like at least a large portion of it. So we have users that look at neurons or bacteria, and when you go through all the different volumes, you can actually see like what's packed inside.
We collect two different types of data. We do single particle data, which we take thousands and thousands of images of a similar protein across the grid. Hopefully it's a homogeneous sample, so the protein's the same. Sometimes we have a heterogeneous sample, but we can work with that with tens of thousands of images. So hopefully that gives us a high resolution structure. Tomography, where instead of imaging the same sample thousands of times, we focus on one particular sample. Maybe it's a part of a cell, maybe it's a liposome or some larger, more heterogeneous structure. So like a cell, not all cells are the same. I mean, all bacteria, they may look the same, but at high resolution, the different parts of the cell are going to be all over the place. So the way we study those is we look at one particular sample, and then we rotate the stage around that sample, and we take dozens of images of that one sample, and we get what we call a tilt series. That tilt series then can go in computationally and turn into a tomogram, where we get a 3D volume of what that sample looks like. And then from that volume, as Miriam said, you can go through and kind of annotate the volume. So you can you mark through like, oh, this is what the cell membrane looks like, or this is what we see a lot of repeating proteins that look like ribosomes or different parts of the cell. And you can kind of start marking them out, and you get an idea of, oh, maybe depending on what your biological question is. We have a group on campus that just published a paper about ferrosomes, and it was about these bacteria that incorporate iron into these little packages. And iron's very electron dense, so in an electron microscope, they show up these big black dots. So it's very easy to see the cell and see these little packages of iron that were just kind of clustered inside these cells. And so they were asking a question about why that was happening.
So it's not unlike, it's different from an X-ray, because of the wavelength, of course, but like the way the image is taken is you're shooting some energy through the object, and you're measuring something on the backside, what comes through. And your ability to tilt this helps you to build out the 3D volume of is that a good way of explaining it?
Yeah. And our usual range is like 60 degrees on one side and 60 degrees on the other. So when you compile all of that together, you get a pretty good picture of what's going on for that one sample.
Okay. And so will you stage it in X and Y and then do the tilt series at different locations?
just depending on, your sample. So if you have your X and Y and say position, your sample number 1 bacteria, you do a full collection there, and then you would move on to the next bacteria that's either in that grid square or somewhere else in the grid.
I see. So you don't get images out of this. You get data, and then the data has to be processed into images. Is that correct?
Images.
We get images.
You get images.
We get images.
I don't believe you.
Yeah.
I don't believe you. I don't believe it till I see it.
Yeah.
So what's fun about this is I don't know a lot of these words, but
We all start somewhere, though, so it's okay.
Thank you. Yeah, you guys you have command of the language, and you said ribosomes. I've heard that before. I don't know what it is.
So this is an image that we collected off the microscope. So this is just a JPEG.
And what dimension are we looking at here? Like, I mean, what resolution?
So this particular this image is 0.7 angstroms per pixel. That's the way we look at this. We look at an angstrom per pixel size. And so the black objects that you see here are protein. This is ice contamination. And then the gray background is just the background ice for the image.
And this wasn't with the screening microscope. This was with...
This is on the screening microscope. And these images look almost identical to what we see off the other microscope. The background's a little bit cleaner on the bigger microscope. Ross, you want to take over?
Nice to meet
We're
you.
at a meeting. We have a grant that we're trying to write for a new microscope, and we're on a time crunch. Sorry.
Well, what are we going to name it? We're going to name it like Mr. Freeze, or
So this one doesn't have a cryo name on it. It's a weird one, but it's a L120C. The C stands for cryo.
the grant's never going to work unless you can name it something cool. It's cold.
Yeah. Ross will help us with that.
Hey, thanks I appreciate you taking. Thank you.
Thank you, everyone.
So how do you take these images? And how do you convert? Well, you showed me the other images. How do you convert that to the model?
So the images, I can walk you through the whole process. And so images here are stored on a hard drive. These data sets are then imported into a program. And these are actually movies and not pictures. These are movies with 50 frames, approximately 50 frames. We use those frames and combine them together. And just like on an iPhone, when we take a picture, which is a live picture, in that live picture, you will see that at the end, there's a motion, but at the end is a nice bright picture with HDR, like good lights. Similar thing here, those 50 frames are put together and matched, and if there's any motion while the image was being conducted, all of those like different frames that are moving are connected together. And that gives us a very nice image. So each of those image or movie now, after doing this motion correction, is now called an image. That image we take and we start picking particles, so like identifying each protein, like, hey, this is a protein, this is a protein. And in this process, we can also use AI. So we use AI, so we pick some by hand and then give the AI that, hey, here what I think is protein, can you go and learn from this and then go and pick it for me? And we can show you in the lab, there's some right now they're running, and it will learn and pick those particles, protein. It will identify a signal from noise. Once we get that, we actually cut a box around it and take those out. So it's like cropping it out, all those particles. And now each particle also has information in it. For each pixel, there is some number to that. And when we start processing, those numbers are put into equations. And each of those equations is run. So we use GPU power like to run all these long equations. And at the final, what we get is an intensity, a CTF, or a contrast transfer function, that we use for further analyzing or connecting those into a 3D model. So once we have these, if you remember, I showed you the images of side view and top view. Once we have those different orientations, each image is taken out and they are overlaid, and the program starts finding different or assigning orientations to them.
So is it like you're finding and it's like a matrix. You see a matrix somewhere, and then there's a shape, and it may be laying on its side like this, or it may be laying this way.
Yes.
You let the computer trying to find, oh, that's a shape I recognize.
Yes.
And then once you find it, then you can start to image based on that, or put that together. Am I understanding correctly?
Yes, you are. And there are two kinds of alignment. One is a 2D alignment, the other is 3D. So 2D alignment is like, for example, taking a picture of my face, 50 pictures of this way, 50 pictures sideways, 50 this way. So there are three classes now, front, side, side. So it will separate those into classes. Now it has some prior information about like, oh, this is one view, this is the other view, this is the other view. Now in the 3D classification, it starts putting them into pieces and assigning them like, oh, I think this will go here. And we do that at different level, meaning we first do that at a low resolution. So for example, if I give you a super zoomed in image of like a puzzle, like there's a beach and there's a house on that beach, but if I want to give you the picture of the window, it would be hard for you to place that where it goes in the puzzle. But if I give you a big picture, you can place it. Same way with low resolution and high resolution. We first start with the low resolution with alignment. So basically, it's just thinks of it as a blob and starts aligning it. Once it has aligned, in further processing, we slowly start giving the program high resolution information, and then it starts moving it just by slight changes, and it's able to align all of this.
So you get to you start with a less computationally intense process.
Yes.
And then you get in the ballpark, and then you hone in and try to refine it.
Exactly. Yes.
Okay.
And then once we're at that point when we have high resolution information, we then go to further more programs that we can run, which is like getting into very nitty gritty, but it's like doing local CTF, running global CTFs, and getting that every single amount of information from these high resolution data sets.
And what was CTF again?
Contrast transfer function.
Okay, got it.
Yeah, and once we get those, we put them together and get a final model. And that model is actually a map, is electron density map. And we put that in the program that I showed, Coot, and we start building the amino acids or protein in there. Now, one thing I did not mention while we were talking was that you asked me a question about like, how do you know this amino acid goes here? Well, one thing was it's a shape. So that shape makes sense. But also, the other thing was that we already know the DNA sequence for these things because we already sequenced it. So we turn those DNA into a protein sequence because we know the protein sequence. And then we know somewhere this particular amino acid has to fit in this chain. So we have a code already. We just have to find a way to fit that code in that density. So it makes it a little easier. It's not all like random reassigning. We have some prior information about the protein sequence of that, and so we use that and start fitting those in the electron density. So it's like a puzzle, and structural biologists love solving that puzzle. That's why a lot of people pick structural biology, because they love this puzzle, like this hard challenge of like, hey, so many people in structural biology field really enjoy by like, hey, I did this was a 4 angstrom map or a 5 angstrom map. So some people will say like, I'm really comfortable up to like 4 angstrom. Anything above that,
oh no, I don't go down to, well, you know, that's amazing.
So like, it's just,
I've got to admit, this is intimidating. Like I've seen a lot of stuff in different fields. This one's complicated. And I feel, because I'm very weak in the area of chemistry, I can feel myself being a little bit intimidated by biochemistry. But the tools are similar throughout a lot of different fields. Like you're taking images and then you're getting perspective views. You're having a computer. As an engineer, we model things and then we make our views, but you're doing it backwards. You get the views and then you're trying to find the 3D model.
That's correct.
It's amazing. So how often do you come over to the microscope lab?
For my project, I come here once, and then if I get a good data, I don't come here for like a year.
So this is where the business happens, but you don't get to hang out here.
Yeah.
Let's take me back to the microscope. So you get the images from the microscope.
So when you get the images from the microscope, they look somewhat like this.
And I can see the little crown-looking thing there. Is that the bottom of a motor?
Yeah, this is the bottom. This is the top view. So I would say this is how it looks. This motor right here is the view of this one.
Okay.
And then there is this view right here is a side view, like something like this.
Okay.
So we click on all of these particles, and once we pick those particles, we go.
So this is the contrast transfer function you were telling me about.
That's right. So this is the contrast transfer function. What we do is the micrograph that you saw, the grayscale image that you saw of the particles, this is a mathematical version of it. So what we do is we crop out small triangles on each of these pictures, and each triangle we give a numerical value, a contrast transfer function. And that is basically digitizing the information so that we can use that to further talk to the program as what we want to do.
You're seeing if images pop off the screen, so to speak.
Yeah, exactly.
Okay.
So then we pick particles, and when we pick particles, meaning we pick those proteins by hand and using the computer, we run a program of 2D classification, meaning we run a program where all these particles that has been picked so far are similar looking particles are put together into classes. And what we see here is some are just junks and some are actually our protein complexes.
So you go through there and you pick the ones that are the good stuff.
Yes, exactly. So this looks like a good stuff. This is a good complex. This is good, but these are not. These are some just crap. So we don't select those, but select the good particles and put them together into a program for 3D modeling now. So we have 2D classes.
Yeah.
Once we have 2D classes, we put them together here to get a 3D version.
Oh, so you build a 3D model, like you make these shapes actually match up.
Yes, each of those classes on pictures that we saw, we start matching them up as to which one is the top view, which is the side view, and program starts matching those up, and it does a very good job at doing that.
So once you get the 3D model, does this just fall out?
Once we get the 3D model, it looks something like this. We can see all sides, and now what we see is a low resolution model. So once we have this is an initial model. We try to collect all the good signals and remove all the bad signals and try to come up with a high resolution structure. So what you see here is an 8 angstrom, and from here 8 angstrom to 4 angstrom, it took us about 2 weeks to get there, 2 to 3 weeks, but we finally got there and got a high resolution structure for this ring.
What does that look like?
So this is what a high resolution and a low resolution structure looks like.
Wow.
So if you see here, the definition, you can see each protein strands, meaning beta sheets and alpha helices, which are part of like that secondary structure of proteins. In the high resolution structure, you can see them. So for example, in the structure here, which is a high resolution, you can see these lines here. Those are beta sheets that you can see.
I see.
But it's hard to see those in here.
Yeah, it's too globby.
Yeah, exactly.
Yeah.
So those are the features that we start seeing in high resolution, and we can start mapping our.
And so you're removing bad data to get to the high resolution.
Get to the high resolution.
And then once you get to the high resolution, then you actually start drawing and mapping the proteins.
Yes, exactly. And that's in the next step. What we do is we load this into a new program called Coot. So what we see here is one of the maps of the complex. So the one at the
4
top,
angstrom map.
The 4 angstrom map. This one actually is even better. This one is 3.2 angstroms.
Oh, nice.
So what I'm showing here is this map that we get. And we are trying to now, once we get this map, we are trying to put the pieces in, the puzzle pieces in, and try to find what protein, what amino acid goes in which place. So since we know the sequence of the protein, or the protein sequence of this protein, we know we have the pieces, we just have to fit it in this electron density. So what you see here is this electron density, and we are going to put, so if you see this curve, there's a curve here.
The coil, yeah.
Coil, and that's alpha helix. This coil, now we know alpha helix, only certain amino acids make that in a certain orientation or sequence would make that coil.
Yeah.
So these prior information helps us trace this puzzle. So if you see now, we can fill these gaps with these proteins.
And this is a shape, a biochemist person would not be intimidated by this shape.
No, that's very common. It's commonly found in almost every protein. Oh, not every protein, but like 90%.
It's intimidating to me. It looks like a bunch of squiggles and but this is normal.
This is normal.
So this is easily interpretable data.
Yes, it is, and it's and if you see right here, for example, there is this extra density coming here. We're like, oh, this looks like there's some protein here that should be going some exact amino acids. So if the resolution is high, meaning we can see these individual chains. If the resolution is low, all of this will look like a blob.
I see.
So because resolution is high, we can be like, oh, let's fill this up with the exact amino acids. So we will fit each amino acids in here. So if you see now, there's amino acids here. There's an arginine here. There's an arginine there, and we know arginine
Did you map these by hand and then just turn the image on? Is that what just happened? Or did you tell the computer to find the shape?
No, we mapped this, or previous researchers have mapped this in the past. We used the information as like, oh, this has been, they have done part of this.
Yeah.
So let's use that and see if that fits in here. If it does, it's good. If not, we go in and do it by hand.
Wow.
So yeah, it can be doing one at a time. It can take weeks to months sometimes, depending on how big your protein is.
But you like it.
Oh yes, this is the best part. This is where like we get answers. This is what we've been like doing all the work for. So like now we come back and sit down, drink our coffee, and we're like, oh, I have now like even when we're driving here, we are so excited. Like, what will I find today? And you come here, you sit down and like putting these, and like, oh, these two bonds are being formed. So for example, I can go here and be like, let me see if there's any bonds being formed. So I go turn on the distances and angles. Sure.
So you're exploring.
Yeah. So now I'm exploring, and I'm like, okay, let me see what is the difference between these two proteins, amino acids. Sorry. And you see there's 2.5 angstroms. So there's a definite interaction between this and this. They're forming some kind of bond. Maybe that is what's stabilizing this complex so well. So like this 2.5 angstrom is a 2.7 to 2.5 angstrom is like a good bond distance to form a strong bond between two residues, two amino acid residues. So this is what gets us excited, that we have found the interaction that are happening in this complex, what stabilizes this. And if we disrupt this can disrupt the motor. This can disrupt the connection it's having or interaction it is having with other proteins.
So once you disrupt this motor,
Yeah.
if you could destroy the motor, or if you could stabilize it, whatever,
Yes.
just if you had control,
Yes.
then you could start to do things that would affect the chemotaxis.
Yes. Bacterial chemotaxis.
Bacterial, you could disrupt the ability for the thing to move where it wants to go,
Yes.
and that's good or bad depending on what you're trying to do.
Yeah, exactly. Like for infection, that's a bad thing because we don't want bacteria to go everywhere in our body and infect us. So stopping the bacteria is almost like having an antibiotic, but not with an antibiotic, because bacteria can get resistant to antibiotic. So stopping them is a secondary way. This is one of the many other options that people.
Maybe you get to invent a new word. Instead of an antibiotic, it's a lethargibiotic. You get to slow them down.
I like that.
That's pretty fun.
That's awesome.
Maybe you get to invent a word, man.
Oh my God, that's a good. I discuss structures, but I think I like that name a lot. That's awesome.
Let me ask you this. You do all this and you see all this.
Yes.
What does this make you feel?
It makes me feel that the work that I'm doing is having an impact on human health and will eventually help my kids have a longer and a better life. I'm hoping that this will help extend our lifespan, at least the following generations that are coming. They probably will live to 120, 130 years. And that's what like gets me excited to like have a better world without a fear of like what can happen tomorrow, whether it's a pandemic, whether it's an infection or epidemic, anything. Those things when pandemic happened in 2020, that was very scary. As a scientist, we come to the labs and we work and we know that these things can happen, but to see them actually happening in front of you is really scary, because you know that there is a possibility that this can happen, but you always thought like it probably won't happen. Like we are working on it, it probably won't happen, but it happened, and that puts another fear. Like if this happens again and happens to my kid or my parents or somebody else, like it's not going to be fun for
So are
anybody.
you working on this from a position of fear?
Curiosity and fear, yes, I would say that. There is a fear, like having an antibiotic resistant microbe going around is a very silent pandemic, as one of the nature editor has put it. And it's silent because we don't realize it, that it's developing, it's happening at such a microscopic scale. We all are not seeing it happening. So it is fear that this can happen, and that fear.
An antibiotic resistant bacteria.
Antibiotic, resistant bacteria. And that is scary, because that's something that if that happens, we need an alternative way to stop that from happening, or alternative way to stop bacteria or harmful bacteria. Obviously there are good bacteria as well, but we need to know how this works, how we can stop them if there is such need arises.
When you look at this cell and you look through all the data and you arrive at a motor that looks like a motor in a car.
Yeah.
What do you feel about that?
My mind is blown when I always knew there was a low resolution structure from a few years ago, and I could see it that there's a low resolution structure, but seeing that such a high detail of these strands and helices and beta sheets, all of those like put together and forms a motor which looks like an electrical motor that we have, just blows my mind, is because we didn't know before building motors that their motors exist, but we humans and bacteria came to the same design. Is just amazing. Like, bacteria decided to make a motor, human decided to make a squirrel cage induction motor, but those both motors look so similar. It's a system that works really well. It works really well, but we are also looking at it at a big scale. We, as you saw, that there is so many images, like 300,000 images. Not all of them are exactly same. They have their own errors, they have their own things in there. Like, some is squishy, some is fat, some has 34, some has 35, some has 36. They have their own errors. All we are doing as a scientist is averaging all of them to have a structure.
I see.
But if we had the capability to look at each image, we would start seeing the differences between all these images. So that's the point where I think that the motor is so interesting, because both bacteria and human making it in their own ways and bacteria making their own way kind of is similar.
The motor structure.
The motor structure.
What's your favorite part about it?
I think the connection between the MS and the C ring. This was made through AlphaFold. So we have not been able to see it in our structures because we were not able to trace it, and that is because that part is in the membrane, so it's hidden, so it's hard to see. But also because the way we solved the structure, we took this part, subtracted it out, and subtracted this one out, so we missed the middle region. So that connection is really interesting to me. But obviously there's a few other things that I cannot talk about right now, but like in our future projects that we are working on, there are interesting things. But right now for me, like these connections just reminds me of my motor that my grandfather worked on, where there's like copper coils that he's putting in these old, rebuilding these motors and putting fresh copper coils, and like it reminds me of that kind of structure.
Because your grandfather was an electrical engineer, right?
Electrical engineer. He used to build and rebuild motors for factories.
Is it fun to know that you're working on motors now, just like your grandfather?
Yes, it is. It's very. It feels very rewarding to be working on something that my grandfather worked in the past. Obviously not the same scale, not the same thing, but just to know that I'm working on motors is fun. It's rewarding.
Prash, how long have you been in America?
I've been in America since 2007. I came to in US in 2007.
Are you working on citizenship?
I am. My green card application was submitted in 2016. It was approved in 2016. Yes.
Yeah.
So my priority date is 2016.
You think you'll get it?
I don't know. The process has been very slow. So it's been 2016. The date. People who applied in 2012 are getting it now.
Yeah.
So I've been in the queue for 8 years. So I think another 8 years, and I think I'll be there 8 or 9 years. So
I'm rooting for you, man.
Thank you so much. Thank you so much.
Thank you so much for all your hard work, man. This is beautiful.
Yeah.
And I love there's something poetic about you working on motors just like your grandfather.
Yes, it's. I tell my wife, it's like this project is so personal to me. I have been all weekend here in this office solving this. Like, there have been days for 3 days I did not go home just because I came in on Friday, I left on Monday, just here working straight. Like, one of the structures that we discovered a few months ago, that structure was solved by the whole group. Like, people, we are purifying protein for a week, we get at the very end, and then I collected data and started processing. All of that was 36 hours of like no sleep, straight from Friday morning 4 a.m. to Monday morning 7 a.m. So it's more than 36 hours, but and no sleep. That's how long it took to get two additional structures for our paper. But that motivation to like get it done, I want to solve this now, and I think this will make a good story because I have a counterclockwise structure. I wanted a clockwise structure to make that story complete. That I knew that my grandfather, if he was alive, he would be really happy to see something that exists like this.
Be proud. I think that's the word.
Yeah, I think he would be very proud. Yeah, so I printed this like, and so that like if he was alive, I would show him because I wanted to see in person too. Like my wife, I can show her stuff on the computer, but like to have it in her hand, it was like, for her, it was like, so this is in bacteria? And like that just face, I was like, this is nice because that gives her a different perspective on things that I'm working on.
Well, for what it's worth, Prash, I'm proud of you.
Thank you.
Thank you very much.
Thank you so much.
So Prash just told me that, Dr. Iverson, you're really good at doing the puzzle. Is that right?
Yes, I'm modeling.
Modeling
on tracing.
So this is your thing? You're good at it?
I love it.
Yeah?
I find it relaxing. It's like a vacation. I just enjoy looking at the data.
That's awesome.
Yeah.
And so you guys were the bulk, the lion's share of the work on the paper?
Yeah. Prash
That's
really
right.
did the lion's share of the work.
That's awesome.
Tina did a lot of writing with me and a lot of model building as well. Yes.
That's awesome.
Yes. And obviously the whole idea and the grant and everything was Tina's idea.
Oh, it's just a little part. That's awesome. They're doing great stuff here at Vanderbilt. All right, I hope you enjoyed this video. A huge thank you to Dr. Tina Iverson and Prashant Singh for just letting me come up and just talk. I mean, this was very little planning, and they just went with the flow, and I really enjoyed the conversation that we had. So big thank you to Vanderbilt University. It's just up the road there in Nashville. I'm in North Alabama, so I really enjoyed this. Thank you for watching this. If you did, I am flattered that you would spend this much time here on Smarter Everyday 2, the second channel. Feel free to subscribe if you're into that. I just put all kinds of stuff on here that I'm exploring. It's really fun. Thank you to everyone who supports Smarter Everyday at patreon.com/smartereveryday. I'm grateful. And also over on the main channel, I have this little section at the end of the main channel video where I talk about like all the stuff associated with the discussions around the flagellar motor. I really like that section because I think it talks about things that come up a lot in debate around this topic. So if you want to, check that out. A lot of people are asking about how to get the file to 3D print this structure, and Prash has a link I'll leave down in the video description. You can go check out his website and see how to do that. Huge thank you to patrons. I'm grateful, and thank you for watching this video. I'm Destin. You're getting smarter every day. Have a good one. Bye.